Hi, am I audible? Hello, can you hear me?
Yes.
Okay, and my slides are visible, right?
No, ma'am.
Okay, let me just share it again. And I have updated the YouTube link, so now you should be able to see the second week lecture also.
Okay, ma'am.
Now, can you see my slides?
No, ma'am.
Okay, I'll just share the whole screen then. Now, can you see my screen?
Yes, ma'am.
Okay, so let's get started with today's session. So this week there are no assignment problems. Sorry, there are no reading assignments. So we have only the assignment problems. So we can discuss everything in detail this week. We have more time. So we'll get started with the questions. You can take like 5 minutes to go through the question and think about it, and then we'll discuss it. Okay, let's discuss this question now. Actually, the first question is at the end, so we'll come to the first question later. The question is, which of the following techniques can be used to measure cell protrusive forces at the leading edge of a cell? So there is a technique called atomic force microscopy, or AFM in short, where you use a very small sized sharp tip to probe the surface, and you will measure by how much the tip comes back, that is the tip deflection, and from that you will be able to measure what is the force uh, that the cell is exerting during protrusion. So the correct answer to this question is AFM. Any doubts? Okay, let's go to the next question. You can type the answer in the chat box, then we can discuss the answer. So the question is, how do cytokeratin D and jasplakinolide influence actin stress fibers? So these are some commonly used drugs that we use in mechanobiology. So we use these to change the structure of the actin cytoskeleton and see what effect it has on the cells. So different drugs have different effects. And so the first one, cytokeratin, it binds to the plus end, that is the barbed end of effectin. So this is the end where new actin monomers are added. So because the drug goes and binds to the positive end, the monomers can't be added anymore. So it destabilizes the actin filament because no more monomer addition can happen. But jasplakinolide, it uh, binds to effectin and it increases the polymerization rate, so it stabilizes effectin. So the correct answer is option A. Let's go to the next question. So this is also about another drug called latrinculin, and this is another drug which binds to the monomers. So the monomers can't go and bind to the effectin, so it inhibits actin polymerization. So both of you are correct. The correct answer is option 2. Uh, when you type the answer, please mention the question number also so that I'll understand for which one you have typed. This question is about cell migration. So, when a cell moves from, like, when a cell goes from one place to another, how it can do that. So, in this, if this is the direction of motion, then the first step is that there is some protrusion in the entire cell, and then this goes and binds to the substrate because the cell is attached to the substrate. And once a new addition is formed in the front, the backside of the cell comes out. And once the backside has come out, this actomyosin contraction happens and the entire cell moves. So these are the four stages. In the first stage, the front side of the cell will be protruding out, and then it will bind to the substrate, whatever extracellular matrix, and then the back side will come out, and then the whole cell will move front. So the correct answer is option A. Let's go to the next question. So this question is about lamellipodia. Lamellipodia is the location in the cell where a lot of cytoskeletal proteins of the cell are present and they exert a force. For example, during cell migration, this lamellipodia is where the force is exerted. So so this is present in the front of the cell. So you have actin and then all the proteins that are associated with actin, like some proteins are needed for the branching of actin. They are present, and some proteins are needed to keep the actin filaments together. They are all present. So the lamellipodia is this structure with all the cytoskeletal proteins present. And just behind the lamellipodia, there is another structure called lamella. So this lamella is where the contractile forces are generated. Lamellipodia is more of a structural feature. So in lamellipodia also force can be exerted, but that will be the force generated from actin polymerization. But the force generated from this actomyosin contractility that will come from the lamella. So myosin 2 is present in the lamella, but in the lamellipodia, all ARP23, coffulin, these actin-related proteins are present. So the correct answer is option 2. Myosin 2 is not present in lamellipodia. It's present in lamella. Let's go to the seventh question. So this question is about Lamellipodia and Lamella. Lamellipodia is what we were already discussing, that all the actin and the associated proteins are present. Uh, and it is in the front of the cell, the leading edge of the cell. And lamella is also present in the leading edge of the cell, but it is just slightly below the lamellipodia. So the lamella is a more stable structure where it is like a reserve of all the actin and other proteins that are needed. But lamellipodia is the structure where it is more dynamic. There is more actin polymerization and depolymerization and retrograde actin flow. All of that is happening in the lamellipodia region. So, because this lamellipodia region is more dynamic, it needs a source for all the proteins to come from, which is more stable. So, that is the lamella. So, just the kinetics of lamellipodia and lamella are different. Lamellipodia is more fast compared to lamella, which is more slow and stable. So they are both present in the leading edge of the cell around where the focal adhesion are present. But lamellipodia is more dynamic and lamella is more stable. That is the main difference. Okay, let's go to the next question. So this question is about actin dynamics. So actin is in the monomer form and it can polymerize into its effectin form. Actually, it's a video of retrograde actin flow, but it's not playing. Anyway, yeah. So it goes from the monomer form to the polymer form. And there is also a backward flow of actin. So this backward flow of actin, why is it coming? It's coming because the myosin fibers are pulling on the actin fibers. So it's something like when you draw water from a well, the rope is going down. So just very similar to that, the myosin is pulling the actin filament. That is why the actin is going back. So this backward movement of actin is what is called retrograde flow of actin. So I will just explain this once. Uh, so actin protrusion is how much the actin filament is growing. So that is actin protrusion rate. So this protrusion rate, if the actin is polymerizing, then the filament can grow, right? So actin polymerization will be a plus sign. But if the actin is going back because of the retrograde flow, then the actin can't protrude, it will go back. So retrograde flow will come with a negative sign, minus actin retrograde flow. But in the question, they are asking for actin polymerization rate. So if you take the retrograde flow to the other side, it will become a plus. So I'm just taking this to the other side. Then this minus will become a plus, and you will get uh, okay, I should have copied. So I have just taken retrograde flow to this side. So this finally it will be an addition. So sum of these two will give actin polymerization. So that's why the correct answer to the eighth question is option D. The sum of actin protrusion rate and retrograde flow will be equal to the actin polymerization rate. Is it clear? I think somebody had typed something else. That's why I was explaining. Oh, both of you have typed option 3, but the correct answer is option 4. This is clear, right? If you have any doubt, you can ask.
Yes, ma'am, it is.
Okay, let's go to the ninth question then. So this question is also about actin polymerization. The monomer form is called G-actin. It stands for globular actin. And the polymer form is F-actin. It stands for filamentous actin. So it goes from the monomer G-actin form to the polymer form. And when it is growing, so the first step is there is a nucleation. So nucleation means some 4-5 monomers have to come together. And once the nucleation has happened to this cluster of monomers, more addition will happen. And as the filament is growing, the two ends are different. So the front end is called the plus end and the back end is called the minus end. In the front end, there will be more addition than deletion, but in the minus end, the deletion will be greater. So when the filament is growing, this will keep happening that some monomers will be getting added as well as in the plus end, as well as some monomers will be getting deleted from the minus end. And finally, it will come to a steady state where the rate of addition will be, rate of addition will be equal to the rate of dissociation. And at this stage, the length of the actin filament will not change. So this steady state is called as actin treadmilling. So the plus end where the actin monomers are binding, this is called the bar dent, and just because of the structure. And the minus end where the actin is leaving from the film F actin, that is called the pointed end. And one more thing is that so how do we know that whether this actin monomer, whether it will bind to the actin filament or whether it will remain as a monomer? So that depends on the how much concentration of the monomers are present. If the monomers are present at greater than a critical concentration, then it will be added to the plus end. Actually, both the plus end and minus end have critical concentration. So for plus end, the critical concentration is lesser. So even if the G actin is lesser in concentration, it can still bind to the plus end. But in the minus end, the critical concentration is very high. So that is why the G actin concentration is not enough and addition doesn't happen. But from the minus end, the G actin is coming out. So the correct answer is the addition happens at the barb end. And option 2 is not correct because in the treadmilling phase, it means it is in the steady state phase and the length of effectant remains constant. And option 3 is also not true because when it is more than the critical concentration, only then the filament can grow in length. And option 4 is not correct. So critical concentration is greater for the pointed end and it is lesser for the barbed end. So correct answer is option A. So these are some of the proteins that can regulate the length of actin. So coffulin can cut actin filaments, and actinin is present between like the individual actin filaments to make a bundle of actin filaments. And profilin is used to so it promotes basically ADP to ATP conversion so that the actin filament can grow. So it promotes actin assembly, and ARP23 helps in actin branching. So this is the last question for today. So this one, both of you are correct. We use a technique called fluorescent speckle microscopy, where only a small fraction. So, for example, if we are using it for actin, only some like 0.5% of the effectin will be labeled with the fluorophore. The others will be normal actin, which doesn't have any fluorophore. So that is why we will get the speckle, because both the labeled as well as the unlabeled actin will come together to form the structures. So because both of them are there together and they move continuously, rather than getting a stable point, we get the speckles. That is why it's called as fluorescent speckle microscopy. So we have discussed all the 10 questions for today. If you have any doubts, you can ask that now. If you don't have any doubts, it's you can feel free to leave the meeting, but I'll be there till late. So if you have any doubts from today's TA session or any other questions from the course, you can ask. I'll be here till late. Assignment problems, we can discuss previous weeks, whatever the answer key has been released. But if you have not submitted it already, then we can't discuss because These are graded assignments, right?
Ma'am, for the assignment number two, ninth and 10th problem, is it possible to discuss it now? The keys are already released.
Yeah, yeah, sure. Can you just type the question in the chat box?
So they have given a graph, and I can read out the question. In a protein unfolding study by AFM pooling experiment, it gives 5 peaks. What is the approximate length change? So this is the question.
And options are
Uh, Options are 0, 20 nanometer, 100 nanometer, and 140 nanometer. Maybe I can share this image.
can you do that? Yeah.
Yeah. Yes, ma'am. Ma'am, I have shared it on the chat. Can you please check?
Yeah, it's just loading. I'll have a look. Hello. Uh, so this AFM pulling experiment, you know, right? So if there is a protein, and depending on how many domains are there in the protein, we will be getting that many peaks. And here they have given the deflection versus the z position, and they are asking what is the approximate length change. So initially the protein may be in a folded state, but in this experiment you will slowly pull the protein with the cantilever tip. It will be a very small tip in the nanometer size range. And uh, the Z is after 120 is the last one. And there are five peaks. Uh, Deflection. This graph, I'm not sure how to interpret it. I'll have a look and get back next week. Is that okay?
Yes, ma'am, sure. Because in the videos, this graph I
Yeah.
was not able to see, so I don't
Okay.
know how to solve the problem.
Okay, okay. Yeah,
And
even I have not seen this kind of a plot before. I'll check
and
by
there's another
again.
question also, ma'am. I have posted those two questions also. If possible, can you please check that also?
Okay, okay.
Because the another graph also it was not discussed in the video as
Okay.
far as I know.
Okay, sure, sure. You have posted one more eighth question also. For quantifying stiffness of a cell, which part of
Yes, ma'am. At that part, I was a bit confused.
huh.
Only indentation. How can you explain me how? Why not another both indentation and retraction?
So for AFM, basically the tip will come very close to the cell, and so you will do something like tapping it with the finger. So like that, the AFM tip will go and tap it, and you can control how much frequency with which you can tap, and like how much up to which, how close to the cell you are going. So uh, retraction is, I think, so if you tap it, so for example, if the tip goes up to some I don't know, a few microns, say some 5 microns, and if the sample is not there, then there won't be any reaction. But if the tip is going up to 1 micron, and if it is touching the sample, then there will be some reaction force and the tip will come back. So that is the retraction. And indentation is, so you will do this. So slowly you will take the tip closer and closer to the sample. And once you see a retraction, you will know that the sample is present at that Z position. So once you have found out where the sample is present, then you will start to press the tip more and more and see how much it can go. So that is called the indentation. And to get the stiffness, so it's basically like touching something with the finger. Then you will know whether it is soft or stiff, right? So indentation is just like that. So you will just touch it with the tip and see how much the tip can go inside. So that is indentation. And so because they are asking just to quantify the stiffness, just this indentation data is enough. So from this itself, you will know whether how much the tip is going inside, whether it is stiff or not. This, I think, this retraction is just to find out where the sample is present. Because when you're doing an
Oh,
AFM, you have to go very close to the sample. That is the concept, I think. This also I'll just confirm again next week.
sure, ma'am. And indentation and retraction in when I search in some papers, they have mentioned we can calculate roughness also, and in some paper stiffness, but I am not sure because I was confused.
Yeah, I think
So that's
retraction,
why.
you can calculate the roughness because retraction will give you like a map of the cell, uh, whether it
Okay.
is in the same Z plane or whether it is in different Z plane. So from the indentation, sorry, from the retraction, you can get the roughness, and from the indentation, we can get the stiffness. I think so. I'll just
Okay,
cut
ma'am. Thank.
again.
Sure.
Yeah, yeah. Any other question? One
Yes, ma'am,
more.
I have put a question.
Let me see. This is from which week's assignment?
Week 3, ma'am.
Week three. Okay.
Yeah.
This one, the answer key is released, right?
Yes, I'll check
Yes,
just in case, huh?
it has.
Sorry, I didn't get you.
It has been released.
Okay,
Yes, ma'am.
okay. Then we can discuss. So this question is about cell migration. Option A is lamella expands outwards while lamellipodium moves inwards. So this is not correct because if the cell is moving front, then the lamellipodia will go to, it will also go towards the front side. It won't come back inside. Uh, lamella expands outwards. Okay, okay. So this question is about the thickness of lamella and lamellipodium. All the branched actin from lamellipodia moves towards lamella in an event called network turnover, which stabilizes protrusion. Option C is not correct. There will be some retrograde actin flow, but even if the actin is going back from lamellipodium to lamella because of that retrograde flow, there will be new polymerization that is happening in the front. So these two will be balanced, and the net result will be that uh, there will be more actin polymerization towards the front. And the actin branching will also be there because the lamellipodium is more like a branched kind of a structure. So option C is not correct. Option D, G actin spontaneously polymerizes in the local swelled part of the cell to produce permanent protrusion. So option D is not correct because this, especially during cell migration, the already this F actin will be formed, and the G actin will go and bind to the F actin that has been already formed. So it won't uh, polymerize spontaneously. It will be recruited to the sites where F actin is already present. So there are we were discussing about proteins like profilin, right? So because these proteins will be bound to the front end of the F actin, the barbed end, so which will make the G actin to go and bind to the F actin which is already present. So option two, lamella expands outwards. So when the cell is moving front, so the lamellipodium will become more dynamic, meaning uh, the cell has to exert force. So the myosin will keep pushing the actin back, and when the cell is, although the cell is moving front, this retrograde actin flow will be that will keep on increasing. So the amounts of actin that is going from lamellipodium to lamella will increase because of the increased retrograde effactin flow. So because of that, the size of the lamella will increase. So I think that's why option two is correct. What? Uh, so in the answer key also they have mentioned option two.
But ma'am, in the
Yes, ma'am.
grading, like the grading page, the answer is written A, but in the answer key, it is written as B.
Okay. Option A is lamella expands outwards while Option A.
No, so in the week 3 updated solution, they have marked the B as the solution.
Yes, yes, but in the graded where we like check the answer now, in that place it's the accepted answer is option A. Can I share the screen like that way I can show?
Yeah, yeah, sure. Because option A can't be correct. Lamellipodium will, if it is going inside, then the cell can't move. Okay, so here they have given option A. Can you complain this with the NPTEL team? Because I am just responsible for the TA session.
Okay,
This
ma'am.
and all, I think you can contact NPTEL directly.
Okay, ma'am. Actually this occurred in previous week too.
This is from, oh, this same thing happened in week 2 also.
Yeah, yeah, yeah.
Okay.
Which one are we supposed to follow? The answer key ones, right?
I think the solution, that Word doc that you shared, that will be the updated one. I think you can follow that.
Okay, ma'am. Thanks.
Yeah.
Thank you, ma'am.
Yeah. Okay. Thank you.